Alright, we're going to resume our bacteria notes and talk about bacteria pili. So these are hair-like structures. It'll look something like that, and you kind of saw in the video yesterday of the intro to cells where it was showing those little bacteria just kind of floating around. They're probably moving with the help of these pili. Pili help bacteria adhere to substances, and then it also allows some exchange of genetic material to happen, and this is called conjugation. And so here's a picture of conjugation. It's just, here's a bacteria extending its pili to another bacteria, and exchanging that genetic material helps increase genetic diversity, which is good for any organism. This is, however, not a true form of reproduction because you're not creating anything new. So it's really important to remember that conjugation is not a true form of reproduction. There's a pili there, just that example again. Another thing on our bacterial surface that we'll see is a flagellum. So flagellum is that long tail-like structure that's off the back of many of the diagrams that you've seen thus far. They are slender, rigid, and helical, and they spin like a propeller. So if you've ever seen a dog get really excited when they see somebody and their tail spins around in a circle, that's kind of what I think about when I think about how a flagellum helps propel a bacteria forward. An endospore is the next thing that you might see on a bacterial surface. An endospore is a thick wall, and it allows a bacteria to survive harsh conditions until conditions improve. So kind of like a hibernation tool, if you will. Here's an example of what a flagellum looks like off the back of a bacteria. These little guys right here are pili too, so it's not that it has to be one or the other. They can definitely have both. And then this is just kind of an example of an endospore where it's got a thick outer covering right there. The three shapes of bacteria, so the first one is bacillus. So B-A-C-I-L-L-U-S, bacillus, and that is rod-shaped. So bacillus is a rod-shaped bacteria, as you can see in this picture. It's rod-shaped. Our next example is a coccus, C-O-C-C-U-S. It is sphere-shaped. So if anybody's had streptococcal pneumonia, that is strep throat. So streptococcus is the bacteria that causes strep throat. Those are sphere-shaped again. Those are in the circles, little circles. And then the last one is a spirillum, S-P-I-R-I-L-L-U-M, spirillum, and that is a spiral-shaped bacteria. So those are the three shapes of bacteria. We only need to know the general shapes. This next slide is going to show you kind of some of the more complex shapes, but this is what you guys will be responsible for. So here's some more of the complex shapes of bacteria. Obviously there's always gray areas, but they're coming off of, basically these all are subsets of the coccus shape, the rod, or the spirillum shape. So bacterial reproduction, this is kind of a misnomer because conjugation is not true sexual reproduction, as it does not actually give you a new organism, but it does swap genetic material. So it's a way for organisms to increase their genetic diversity. Binary fission, so this is how bacteria reproduce. The term is called binary fission. You definitely need to know that. And this is where a circular DNA reproduces and the bacteria itself splits apart, and this is considered asexual reproduction because you are getting a genetic complete copy from the parent to the offspring cell. So here's kind of an example of mid-binary fission of two bacteria cells. So these will be identical genetically to each other. Bacteria picking up DNA. So bacteria can increase genetic variation by picking up fragments of DNA. Ways that they can do it is by conjugation. So they can exchange DNA with another bacteria through pili. They can have transduction via a virus. So this is just a process where DNA is introduced to a cell by a virus. So that virus, and we'll talk more about how this happens when we discuss viruses next week or the week after. And transformation, which is free particles from dead bacteria. So transformation is just simply a direct uptake of DNA particles by some bacteria. So that's how bacteria can increase their genetic variation. And we talked in class about genetic variation a little bit, but the more genetic variation you have, the harder for disease it is to get wiped out by a disease. So bacteria, the one reason why we can fight it with antibiotics is because most of them are genetically identical to the other. And so we can get a set of antibiotics and kind of wipe them all out. Genetic variation allows for that organism to better survive those changes. All right, so how do bacteria eat? So metabolism, autotrophs. Autotrophs means self-feeder. So auto is self, troph is feeder. So some organisms are photosynthesizers. So photo is from the sun, synthesizers using organic molecules, and it's going to use the sun to create its energy. So we've got two types of photosynthesizers, aerobic photosynthesizers. Aerobic means with oxygen, so it's going to be organisms that use oxygen and the sunlight. And then anaerobic, which is things, anaerobic is without oxygen, and it uses sulfur or acids to help create energy, but no oxygen. Another example is chemoautotrophs. So chemoautotrophs use inorganic molecules, chemicals. Chemo is chemical, autotroph is self-feeder. And so chemoautotrophs are organisms that use chemicals to create their own energy. Okay, so photosynthesizers and chemoautotrophs are two examples of how organisms eat. Another example is a photoheterotroph. So photo is sunlight, so it uses light for energy. Hetero is opposite or different, and troph is to eat. And so it just means that it needs to eat something else in order to get energy. It's not going to create its own energy. So for example, plants are not photoheterotrophs, right? Because they make their own energy. So plants are photoautotrophs. A photoheterotroph is going to use light for energy, but also must consume organic matter. So anything that's eating something is a heterotroph. A chemoheterotroph is something that it cannot make its own carbon. It cannot use its own carbon, whereas a chemoautotroph can. A chemoheterotroph is one that uses chemicals and it ingests chemicals to make and create its own energy. Okay? And so it's anytime you see hetero, you need to know, heterotroph is that you need to know that it's ingesting or consuming something outside of what it is in order to obtain energy. Bacterial respiration, so aerobic bacteria, so aerobic is with oxygen, so aerobic bacteria must have oxygen to survive and reproduce. And obligate anaerobes live in the absence of oxygen and in fact cannot live in oxygen environments. So this was really fun to study in my microbiology class in college because we actually got to grow some obligate anaerobes where we were piercing our agar, so you guys will learn all about what agar is next week, and actually getting bacteria to grow where we normally wouldn't be able to study that bacteria on surfaces or anything like that because on surfaces of things that we see all the time have oxygen available. So it's just definitely interesting to learn about. So again, obligate anaerobes live without oxygen, and then facultative anaerobes can live with or without oxygen. So these guys are kind of the go-between between aerobic and anaerobic bacteria. So facultative anaerobes can live with or without oxygen. So good and bad bacteria. So obviously we want to make sure that we're brushing our teeth and having good dental hygiene because bacteria in our mouths can definitely cause some problems. Diphtheria is an example. Dental caries is an example. Okay, so it has on your notes good and bad examples of bacteria, so you'll want to write some of these down. Conjunctivitis, which causes pink eye, so that's definitely very contagious. So if you've ever known somebody with pink eye, they've probably been homebound for a little bit so that they don't spread that disease. Botulism, so this is why canning is so important, that you heat everything up so that you can kill the bacteria. But botulism is also is used to get rid of wrinkles. So botulism is a nerve toxin, and you'll often hear it called Botox. So people actually inject this nerve toxin into their face so that they don't have wrinkles. I don't know why you would do that. Wrinkles are beautiful. Lyme disease is another example. So Lyme disease is carried by ticks, and this is actually a bacterial disease. Black plague is another one. Black plague is carried by fleas, and this is just a bacterial infection that causes some death to the skin cells, so that anytime you see those black tissue, that's tissue dying, so that's obviously not good. Good bacteria. So there are good bacteria. So any bacteria that's in your gut is really good, and we can help maintain stable bacteria in our gut by our diet. So if you've heard before that yogurt has bacteria in it, it's definitely correct. Yogurt has really good bacteria to help us continue to maintain that healthy gut. So there's good microbes in our tummy. Lots of food preparation also includes bacteria. Sauerkraut, wine, vinegar, yogurt, and chemical manufacturing also include good bacteria and use good bacteria. Nutrient flow. So major bacteria break down and recycle nutrients, and this is really, really important. So bacteria help recycle nutrients in our atmosphere. So there are decomposers. These are organisms that decompose dead organisms. And if it weren't for bacteria that decompose it, we would be sitting on a pile of dead things all the time because there would be nothing to get those to go back to their carbon-based life form. So if you think about in the forest, when leaves fall and when a tree falls and when organisms die, bacteria help decompose that and kind of recycle that nutrients. If we didn't have that, those organisms would never go away. So we need those bacteria to be able to do that. Sewage decomposition, there's bacteria that convert waste into usable substances, so that's also really important as well. So there's important in nutrients flow. Nitrogen recycling, so bacteria live in the roots of legumes. Legumes, potatoes, or beans are examples of legumes. And it takes atmospheric nitrogen, so these bacteria take atmospheric nitrogen and change it into ammonia and nitrates. And these are usable forms of nitrogen for plants. And that's really, really important because nitrogen in and of itself, so N2, nitrogen, N2, is not usable for plants. But nitrates here and ammonia here are very usable for plants. So this is just, that's an example of a legume there. That's where, right here, and then these nodes are where those bacteria are going to live. And this is just an infographic, and you have this on your notes, of that cycling through of bacteria forming that nitrogen. We call it nitrogen fixation. So it creates nitrates and nitrites and ammonia, so different versions and forms of that nitrogen that are useful to plants and animals. And we'll go through this more at a later time as well, but it's important to understand that there is such a thing as nitrogen recycling.